Okay, well, it's really a privilege for me to be able to introduce this morning's speaker, Yoni Kipnis. I've really enjoyed following his career over the last 15 or so years after I moved into the area of microglia biology. And so we have a real treat today. So not on his CV that I got. I do know that Yoni spent his formative years in Tbilisi, Georgia, where there was there's a significant history there in glial biology that occurred in the 70s through the 90s. And Yoni organized a meeting in Tbilisi in 2019 on the topic of functions of neuroglia. And so I had the opportunity to visit and participate in that meeting, and that was a lot of fun. So from Tbilisi, Yoni went to Israel and obtained his bachelor's in biology at Tel Aviv University, and then he went on to do PhD and postdoctoral training in neurobiology at the Weizmann Institute. And there he worked with Mikkel Schwartz and initiated his interest in studies in the intersection of the immune system with the brain. He had his first independent academic job at the University of Nebraska from 2005 to 2007, and then moved to the University of Virginia, where he accelerated up the academic ladder, and he formed the first iteration of his big institute on brain immunology and glia. I think he's had many contributions, but I think the one to highlight for today was the report of his laboratory in 2015 of the previously unknown structural and functional features of central nervous system lymphatics. And that was really a huge breakthrough finding. It really changed the way people were thinking about the brain as an immune privileged organ system. And it has sparked huge interest in many subsequent studies. And so I think that will be the focus of his talk today, and we'll look forward to hearing the latest developments. Notably, Yoni moved from the University of Virginia to WashU in 2020. He's currently the Alan and Edith Wolf Distinguished Professor of Pathology and Immunology and Professor of Neurology, Neuroscience, and Neurosurgery at WashU, and he moved his Center for Brain Immunology and Glia big. So that's still big at WashU. And Yoni, we look forward to your presentation.
Thank you so much, Chris. This was too nice, too kind. Thank you. Okay, so let me share my screen with you, and I think you should be able to see it now. Yes, and a mouse.
That's good.
Okay, great. So because it's ADRC, I had to present some of the Alzheimer's data, even though we have something new which will come at the very end. But anyway, so the first part is published, and then I'll show some maybe less related, at least on the surface, to AD, even though I think it is very related to AD. And then I'll show some unpublished data, which just got recently accepted, which I don't know, I'm really truly excited about. So anyways, the first part is published, and then towards the end, it will be more unpublished, and I think some very interesting, at least very, not maybe very interesting, but very intriguing findings that I'll show you. Let me maybe start with my disclosure. A very brief intro, and I think people know there's the two systems. Neuro-immune have been believed to live in disconnect and only interact when there is a problem, when there is a pathology. And I think it all goes back to this idea that CNS is immune privileged organ. And of course, when you create a definition like immune privilege, and then you ask 10 people what it means, then you get 12 different answers. Everybody probably understands it in their own unique way. And then one of the dominant understandings was that immune system has nothing to do with the brain, and that's the only way the brain is able to actually function properly. And if immune system is involved, then there is a problem. So this definition of immune privilege actually dates exactly 100 years ago to June of 1921, when Japanese scientist Shirai published a very, very small paper in a Japanese medical journal, showing that if you take sarcoma cells from rats and you implant them into brains of mice, they will not be rejected. And of course, they'll be rejected anywhere else in the body because they're coming from another animal. So this was kind of the first demonstration that there is something unique about brain immune system, or at least the first demonstration that I'm aware of. With the eye was a little bit earlier, but with the brain, this was the first paper. And then 2 years later came a much bigger paper from Rockefeller University, but showing pretty much very similar things, is that the brain will not reject tumors at all, whereas in the periphery, they will be very easily rejected. And then 20 plus years later, Peter Medawar, of course, doing his experiments on skin transplantation and understanding the tolerance, for which he received Nobel Prize later on. And he actually made these skin transplants also into the brain and showed that if you take a non-matched controls and you put them, of course, in the periphery, they will be rejected very efficiently. But if you put this, let's say, if you take two mice, black and white, even though he was doing with rabbits, but let's say mice, black and white, so you take a tissue from a skin from a white mouse and a black mouse, immediate rejection. But if you put this white skin into the brain of the black mouse, it will not be rejected. It will stay there for a very, very long time. And I think one of the things that he did was very exciting and very interesting is if you take a skin from a white mouse, put it on the skin of a black mouse and in the brain of the black mouse, then you will get rejection on both sides, which right there today tells us that, well, there is probably activation and then the cells can get into the brain. And back then, it was too much to digest. So the second part of his work was almost ignored. And this idea of immune privilege kind of really synced really well. And the mechanisms were that the blood-brain barrier is a mechanical barrier which prevents immune cell entry into parenchyma of the healthy brain, even though we know that if there is a virus in the brain, immune cells will get there. We know that if there is an ongoing inflammation like in MS, we have immune cells coming in there. We also know that Medawar showed, right? If you activate cells in the periphery and then you put them tissue in the brain, they will go. So blood-brain barrier probably is not really as impermeable as we thought it would to be. Also, Tony Weiss-Coren now shows beautiful data that actually even molecules from peripheral blood can move across blood-brain barrier. And the other underlying mechanism was that the CNS has no lymphatic vessels, and therefore there is no direct and physical connection with the lymph nodes and the immune system, and so there is no way to inform the peripheral immune system about what's happening in the brain. And the second part we showed to be not really, to be only partially correct. So there are no lymphatic vessels indeed inside the brain parenchyma, even though there was the one really embarrassing paper a few months ago in PNAS from a group at NIH, I forgot the last author, claiming that there are lymphatic vessels in the brain, but they were using a marker for LIVE-1, which labels perivascular macrophages. So it was really wrong. So nobody, I think, so far has seen, at least in physiology, lymphatic vessels inside the brain parenchyma, but it doesn't mean that there is no lymphatic connection between the brain and the periphery. And so that's what I'll show you some of our older work. Just maybe to give, I'm sure this most of you are probably neuroscientists, but just to kind of put people who are not on the same page with all the rest. So we have brain parenchyma. It just where the main immune cell, of course, is the microglia, and just outside the brain parenchyma, we have between the brain parenchyma and the skull, we have the tripartite tissue called the meninges. Collectively, it's made of 3 layers. The pia mater is attached to the brain. Then we have a dura mater attached to the skull. Underneath dura, we have arachnoid, and then between the arachnoid and the pia mater, this is where CSF flows. And of course, what was really exciting to us is that when we looked into this area, while the brain really immune cells there are mostly microglia and perivascular cells, when we're looking into the area which is surrounding the brain in meninges, then you can find all kinds of immune cells you can only think of. Basically, if you compare the composition, the diversity of immune repertoire in the meninges, it is way closer to what you would find in peripheral tissues or barrier tissues like gut, lung, and maybe even skin. And then, for example, if you compare it to diaphragm, so it seems like it's a very, very well populated by immune cells. And of course, the question is, what are these immune cells doing in this area? So my lab is very interested to understand the contribution of different immune cells and their cytokines on brain function. So we, for example, show the different immune cells through their derived cytokines actually can directly affect behaviors of mice, targeting receptors for those cytokines expressed on neurons. And so some of this work is published, but I will not be talking today about this because this is kind of, I wanted to talk about something else. So that's the immunology of the brain is there, and just in the periphery, not inside the brain. And I will not be talking about this whole concept of, basically, maybe just mention it briefly. What we believe is happening is that instead of having immune cells come and patrol the brain from inside, like they do in any other organ, the brain actually takes all of its antigens and brings them out into this area of meninges where they're being surveyed by the immune system. And so we had a paper earlier this year where I think we demonstrated this to some extent, and of course there is some more work to demonstrate this even more convincingly. So today I want to talk about a little bit about lymphatic vessels, at least in the first part of the talk. And so every tissue in our body, of course, is vascularized by blood vessels, and will at least, so everybody on the call, of course, knows at least something about blood vasculature. But every tissue also vascularized by lymphatic vessels. And unfortunately, probably we have some people on the call who don't know anything about lymphatic vessels, like I was, for example, just a few years ago. And if you think of your household and you have the water pipes that bring all the goodies into the water into the household, or oxygen, glucose, and nutrients into the tissues, and then you have your sewage pipes which remove all the waste. And so this is what lymphatic vessels are doing. They're removing all the interstitial fluid from the tissue. They also patrolling so the immune cells are usually leaving the tissue via lymphatic vasculature. And so the question is, okay, if these vessels are removing all the waste from the tissue into the centers, the lymph nodes. So just think of your neighbors going through your garbage can, right? They will know everything about you. And if there is something unusual in your garbage can, they will say, wait a second, something is going wrong in that house. So same thing with the immune system. They're sitting in the lymph nodes and going through the waste coming from the tissue, and unless they find something unusual, like for example antigens which belong to viruses, or some new antigens maybe induced by cancers, etc., they don't respond. But if they find something abnormal and unusual, then of course they go into the tissue, patrol the tissue, and see what's happening, what's going on there. So the question is, if we don't have lymphatic vessels inside the brain tissue, how does the brain communicate this important function? How does the immune system survey the brain? So, and how does the brain remove all of its waste products? So a few years ago, Mike and Nidegard came up with this very interesting, comprehensive idea. She put together everything we knew and added a lot of mechanistic insights, and then proposed this system or conceptual framework to understand how fluid washes through the brain. And she called it the glymphatic system. So the G stands for glia, and the lymphatic stands for lymphatic function, even though there is no lymphatic vessels in her system. So the glymphatic system means that the CSF will dive along with arteries into the perivascular space that arteries are forming due to glia limitans here. So the glia limitans, hence the G for glia. And the CSF will dive in, and then because of arterial pulsation, CSF will be pushed out into the parenchymal of the brain, and will create this convective flow, which there is a lot of argument whether this is really convective or not convective, but I think for the sake of the argument, it's not really that important. What kind of flow is that? And then it will be recollected at the perivenial space and then be removed back into CSF circulation. So clean CSF comes in, washes through the brain, comes out as the CSF, and then is being washed out. And so here, on the way, what's depicted here is that it collects this, for example, amyloid beta on the way out. And the question is, how does dirty CSF then leave the cranium? And of course, we know that we produce about 3 times the volume of CSF. So we make roughly about half liter CSF a day. So where does the CSF go? So what we know from textbooks is that CSF is going through arachnoid granulations directly into the sinuses, which are the major blood vessels at the top of the head. So directly there. So this actually has been questioned recently by a Norwegian group in humans, and also by us in mice. And so we don't think that CSF is really being washed into the sinuses, but I will not be again talking about this today. What was proposed for macromolecules and for immune cells is that they would actually go all the way towards the cribriform plate, where olfactory nerves are coming out into the nasal mucosa. And then from there, they will be going into this afferent lymphatics. So basically the nose and the brain will be draining through the same kind of same channels, something that doesn't really make much sense. The problem with us was that, not that we questioned this route, because I'll be very honest, 7-8 years ago, I didn't care much about this route, because I was taught that T cells, immune cells, don't leave the brain, and therefore, there is no such thing as really presentation of brain antigen somewhere in the periphery. So this idea never bothered me. But we were interested to understand how immune cells are coming and going into this area of meninges, and the problem was that we could never see any immune cells in this area. But if we peel the meninges, then we would find a lot of immune cells there. So Antoine Levaux, who was a postdoc in the lab, developed this whole mount of meninges. So that today Antoine is PI in the clinic. So he made this whole mount of meninges, and then so what you can see here, this in this nice vessels, those are the sinuses that are draining all the venous blood from the brain. And so we find a lot of immune cells there, which I'll show in subsequent slides, so I'm not showing it here. But if you label this for immune cells, you'll find lots of immune cells. And what was interesting is that when we labeled with one of the markers that's supposed to label actually some of the perivascular macrophages, LIVE1, we found structures that looked like vascular structures, and we said, okay, so what are these? These are not cells. These look like vascular structures. So then, we labeled them with other additional markers, so PROX1, podoplanin, and VEGFr3. So these vessels are really bona fide lymphatic vessels expressing all the right molecules that they should express. So in the eye, for example, we have Schlemm canal, which functions as lymphatic vessel, but is not really made of lymphatic endothelial cells. And so here we have these vessels outside of the brain, but very close along the sinuses, which are actually expressing all the lymphatic markers. So the question was, okay, are these vessels draining CSF, yes or no? So to answer the question, we made a window in the middle of the brain, which of course, those of you who are making windows know that usually go on the side, not to damage the sinus. We had no such luxury. So we had to do this window in the middle of the head, and we injecting now with 2 types of tracers. One green tracer goes intravenously, so all the vasculature is labeled green, and the other tracer we're putting into the CSF. And then after some time, what we see is one vessel next to the sinus is being labeled with a tracer from CSF. And so this is a live video. You can see the flow of the fluid through the flow of the green tracer through the sinus, and the flow of the CSF tracer through the this lymphatic vessel. So anyway, if I summarize any lymphatic, what's happening? Oops, sorry, 1 second. I don't know what's happening with me here. What's happening? Okay, 1 second. Can you see my screen? Can you see my screen?
No, now it's back.
Yeah,
is it back?
Yep, it's back.
Okay, sorry. Anyway, so every lymphatic bed is composed of 3 sections. One section is what we call initial lymphatics, and then we have collecting lymphatics, and then we have destination, which is a lymph node. So if you think of when you're going to work, you're first driving with your roads within your town, then you take the highway, and then you get into your destination. So for the brain, these initial lymphatics will be here at the top of the head, and those are the lymphatics which probably drain all the CSF very easily, because the connections between the vessels are very loose and there is no smooth muscles around. Then the next stage is this highway, where once you are there, it's just going from point A to point B. There is no exits in the way. And so we believe, based on ours and other works, is that somewhere around ears, that's where smooth muscles are starting to appear. And we're also seeing valves which will not allow a backflow of the fluid. And so from ears down, this becomes more collecting lymphatics. And then it gets into the lymph nodes. And so this is Zach, who's in the lab, made this beautiful video where he puts tracer in the CSF, and you can see how slowly, and he opens the neck and images these draining lymph nodes, and you can see how nicely the lymph nodes is slowly being filled with the tracer. So after we published our paper, was a few weeks later came paper from Italo showing basically pretty much very similar findings. Then there were a series of 3 papers from fish. Therefore, these vessels are evolutionarily conserved. So actually, it was 3 different continents. It was from Europe, from US, and also from Australia. Was a paper in rats, and then was also from Danny Reich's group was paper in humans and non-human primate, showing that the vessels exist in those animals and in humans. And interestingly, the structure is not much different from what we see in mice. So I'm kind of now cutting all this immunology related stuff, and I'm getting to the Alzheimer's. And the question was, are these meningeal lymphatics really playing any role in aging and in Alzheimer's? And so this is the work that was done by Sandra Damashkira in the lab, who is now PI at the Mayo Clinic, really talented guy. And so what Sandra was asking is the following. So if this lymphatic vessel is really draining macromolecules from the brain, will this also then allow perfusion of the brain? So in other words, if the lymphatic vessels are not functioning and then there is a buildup of a dirty CSF, if you wish, will this interfere with the perfusion or with the lymphatic flow through the brain? A reasonable question to ask. And to make a long story short, he actually showed this is exactly what's happening. So this is just one example I'm showing you here. We're taking a mouse, we can do different ablation of lymphatics. None of them is perfect, but this ablation is the one that we're actually doing. This is so this here we're killing the vessels using a pharmacological agent, and then we're injecting them with gadolinium into the CSF, and we're following the flow of gadolinium through the tissue. And so you can see that the different sequences, you can see the perfusion of the control mouse brain. This is particularly looking at dorsal hippocampus, but we assessed some other areas as well. And if we're looking into the animal that got ablation of this dorsal lymphatics, and you can see that the perfusion through that area is substantially reduced. And so again, this has been shown in many different systems and in different transgenic mice. So it seems to be a quite reproducible finding that without functional lymphatic, we're also having impaired perfusion through the brain. Now, in old mice, it's been shown that perfusion is impaired. So we're going to look into lymphatic vessels. So lymphatic vessels in old mice are pretty impaired. So there is a less coverage of lymphatics. They probably degenerate with age. And we're showing also a significantly impaired drainage, the function of those lymphatics. So if we put tracer in the CSF, much less of it will come here, comparing green area to green area, so much less of it will come out into the periphery. Nothing is new compared to other tissues, because in other tissues also the function of lymphatics with age is being impaired. So now we said, okay, well, we know that in aging, perfusion through the brain is also impaired, and that's been shown by many other labs, and here we're reproducing. So here using another method, when we take a tracer, putting into the CSF, and then following it by, obviously, killing the animals after different time points, and we're following the amount of tracer within the brain. And so you can quantify it here, the area covered, for example, in this case by fluorescent albumin, and you can see that in animals that are old, very little CSF getting into the brain. So then we said, okay, we don't know how to fix this, but we can fix the lymphatic function. So by impairing lymphatics, we're impairing the flow. Can we maybe improve lymphatics and then improve the flow in those old mice? So we're taking the mouse and we're applying them with a gel just on top of their heads, which has recombinant VEGF-c, which targets primarily lymphatic and endothelial cells. As you can see here, vessel grows, becomes wider in diameter, also covers more area. And so it's showed here. And quite nicely, actually, the function of these vessels in old mice is very much improved. They become almost as functional as they are in young mice. And now the question, will this be sufficient to improve the perfusion through the brain? And the answer is actually yes. So here we can see control animal perfusion, very basic perfusion, which is very, very poor. It's an old mouse. We would treat them with VEGF-c. We actually see a much better perfusion through the brain. And this correlates with a slightly improved cognitive behavior in several behavior tests, and more of using of the hippocampus, which is quite nice. So we thought, okay, we solved the problem of Alzheimer's. We take Alzheimer's mice, treat them with VEGF-c, and the problem is solved. We cured Alzheimer's. Well, the only issue was that nothing worked in animal models. And so we couldn't understand why. it works so nicely in old mice, and it's not working at all in Alzheimer's mice. Because the idea was, okay, well, we enhance lymphatic function, and so all the amyloid beta will be taken away, and it will be all wonderful and great. Well, unfortunately, it didn't happen. So we decided to kind of go back and understand what's happening. So actually, this picture I took when I was visited UCSD last time, or one before last time. I was in Bill Mobley's office, and he told me that actually I did not know that. It's a shame, but I wasn't aware that the first, you can actually see his shades of the window from his screen when I was taking this shot. So he was telling me that he told me that actually the first amyloid was isolated from human meninges. So the amount of amyloid in the meninges is quite high. And indeed, if we're looking into patients, what we see in Alzheimer's patients, if there are plaques in the brain, there will be also plaques anywhere throughout meninges, in the dura, in the leptomeninges, in choroid plexus, everywhere. Now, what's happening with the mouse? If we're taking 5-XFAD mice at about 5-6 months of age, we of course see lots of plaques inside the brains. But if we're looking into the meninges, we actually do not see plaques almost at all there. And then it dawned on us, we said, okay, we're working with young mice, so the plumbing is all good. So we are overproducing amyloid beta, but the plumbing is still functional. So there is no way we can improve this perfectly functioning sink, right, to remove these plaques. But in a real disease, it's not only about the production; it's also a lot about clearance. And so, in old patients, of course, we know that it is likely, at least in old mice, we see deterioration of lymphatic vessels. So we said, okay, let's first of all ask ourselves, if we take a mouse and we impair lymphatic function, what will happen? So we take a mouse, young 5-XFAD mice, we kill their lymphatics using our photoablation therapy. And what we see is in the mice, so we have two controls, we have drug only and the laser only, and then combined together, we ablate lymphatics very efficiently. And what we see now is we see a big substantial buildup of plaques in the meninges, and also more plaques and larger plaques inside the brain parenchyma. So dysfunctional lymphatics will cause accelerated pathology in animal models. And then we said, okay, well, if the sink is not functioning and we bring these antibodies, will they be even able to work as properly in a model or in an organism that doesn't have the sink? So they, of course, may be able to get into the brain and maybe even dissolve some of the plaques, but then all the oligomers, which are quite toxic, how will those be removed if really the sink is not functioning? So what we did is we took the mouse, we did ablation of lymphatics, and then we treated the mice with anti-amyloid antibodies. So we used two different types, aducanumab and I forgot which was the other one. And so we got very, very similar results in both cases. So first of all, of course, when we ablate lymphatics, we have more plaques, which is nice. We reproduced ourselves. And then when we treat these mice with aducanumab, we see quite substantial removal of plaques. There is some more plaques left, but that's okay because we start from a higher level. So there is the removal of the plaque is maybe again, maybe there is more plaques left, but the plaques are still being cleared quite all right. But then when we look deeper into the tissue, what we see is we see a quite abnormal activation of microglia. So microglia that respond to removal of the plaque in an animal that doesn't have functional lymphatics seem to be much higher activated than microglia in the animal that were removing plaques and have functional lymphatics. So this to us was very, very interesting. And we decided to look a little bit deeper into this microglia, which we get is a result of this plaque clearance, but inability to remove whatever metabolites are being formed. So we compared microglia from these two conditions, and we actually were surprised. It came with a lot of interesting changes in many genes. Of course, the depth signature was altered and whatnot. Lots of pathways were altered. And again, the only difference here is that the animal does or does not have functional lymphatic vessels. So otherwise, it's Alzheimer's mice with or without anti-amyloid therapy. So because we are now at WashU and we have this amazing collaboration with WashU folks, including the Diane people, who you may know Diane is a dominantly inherited Alzheimer's network. So they have patients that actually have the actual familial mutations. They have patients without sporadic disease, and then pre-symptomatic cases as well. So that's autopsies that showed plaques, but there was no real disease phenotype in these patients. And then there was also patients which are age-matched but do not have plaques or do not have dementia. So we have now 4 groups of patients, which of course is very interesting. So and what was beautiful is that a microglia has been sequenced from all these patients, and so we have I forgot the number, but it's close to 100,000 cells. So it's really a quite substantial amount of microglia. And so here we are doing, so we're looking at these 4 groups of humans, which, by the way, something very interesting. You may see this at cluster 4, for example, of microglia, which we're still looking into it. You only see it in familial Alzheimer's disease. You do not see, you see very, very rare cases with sporadic in here. So you only see them, or mostly see them in familial disease. And so then we took our 2 mouse groups, which is with ablation or without ablation, and we did integration of human and mouse data together. And so what we're showing with this, so here we can actually look through all 4 different conditions in humans. So in green are the homeostatic microglia, and then in pink and in orange are activated microglia phenotypes. One of them is MHC, the other one is more interferon or vice versa. So you can see that if we're looking into mice, what we observe is that when we ablate meningeal lymphatic, we're losing homeostatic microglia cluster, and we are enriching for the clusters which are activated. Now, if we take these genes, the difference between these 2 microglia clusters is about 60 genes. If we take now those genes and we blast them against human genes, they are primarily enriched in cluster 2 and 3 in humans, which are again activate microglia. So it seems like, and again, this is a lot of hand waving here, but what this paper, what we're suggesting in that paper is that when lymphatic are dysfunctional, it is pushing microglia towards more aggressive, more highly inflammatory phenotype. And we actually, in what happens in mice is when we ablate those lymphatics, and microglia now become a little bit more similar to what we see in humans, or at least the difference in gene expression overlays with those clusters of microglia, which we see in activated clusters in humans. So then we asked ourselves, okay, VEGFc alone is not working, but how about combination therapy? What if we provide this anti-amyloid therapy, but we're also improving the lymphatic function? So altogether they function more efficiently. And so first of all, it was nice to see that even on the plaque level, we were able to clear plaques a little bit better in 3 different mouse models. So these are young mice, 5XFAD mice, where we combine VEGFc therapy plus anti-amyloid therapy, but also in 2 other models. So this is ABP Swedish mutation with really old mice, and so here again combination therapy leaves less plaques, and these are all J20 mice about 1 year of age. Also combination therapy leaves less plaques, but again the plaques is not the most important readout, because we know that in humans the plaques are being cleared, but of course there is no improvement on other aspects. So we actually looked at other aspects of brain physiology, some behavior. But so what I'm showing you here is that when we provide this combination therapy, we can we improve blood-brain barrier integrity, or preserve microbiome integrity. We have less dystrophic neurons, and we have less activated microglia. So it seems like improvement of the, so basically unclogging the brain sink allows more efficient function of this anti-amyloid therapy. What we also showed in this paper, and it's not here, is that some of the SNPs which are linked to Alzheimer's disease are in the genes which are quite highly expressed on lymphatic endothelial cells. Unfortunately, we don't have transcriptome of human brain lymphatic endothelial cells, but we have that of mouse. And so some of these mutations, SNPs, which are linked to Alzheimer's, are targeting those genes as well. Also, some of the main genes which we are all interested, like such as, for example, ApoE, is expressed not only in blood vessels and in microglia, but, for example, also in lymphatic endothelial cells. So it is possible, the dream is one day that maybe we will be able to combine therapy where we enhance lymphatic function plus amyloid therapy to get a more efficient clearance of the plaques. But maybe before then, what would be very, very interesting if there was a way to predict which patient has lymphatic dysfunction based on their SNPs and which doesn't, and maybe tailor this immunotherapy, at least for now, until we have combination therapy towards patients that do not have functional lymphatic impairment, and so they may be able to respond to immunotherapy better. So this is the Alzheimer's part of the talk, and now I would like to switch, unless there are some urgent questions which you would like to discuss now, and then we can do second part, or I can do second part, and then we can cover all the questions together.
Well, I guess I'm your host, so I get to take the prerogative of asking a question.
Okay.
So, I mean, as you know, we've been very interested. I think you were just alluding to, trying to figure out how genetic risk alleles, operate and how they might operate on this system. And I think we've been interested in trying to line up non-coding risk alleles with enhancers in various cell types, and there's a lot of missing enhancers. There are, I guess what I would say is there are a lot of risk alleles that don't fit into any of the enhancers that we've found in neurons or astrocytes or microglia. So there must be other cell types in which these risk alleles operate. I mean, do you have any thoughts about that?
So, Chris, it's a shame, right? When I'm saying it now, and I admit to it, and it's really a shame. We have tried many techniques, and we're now trying some new ones to isolate lymphatic endothelial cells from human patients. So to this date, there is no LEC, lymphatic endothelial cell transcriptome from humans, at least so from periphery, yes, but not from the meninges. So we do not know what they express and what they look like. I think this is absolutely critical information to have their DNA and their RNA. So we do not have that. So I guess without knowing really what LECs are expressing, what the transcriptome looks like, we probably can only make these predictions and I would say unsubstantiated claims. But of course, I would love to believe that maybe some of the missing cells in your system are lymphatic endothelial cells. In mice, I think in mice the data is very, very strong. With functioning lymphatics, we get much more efficient therapy, and with dysfunctional lymphatics, we get a much less efficient therapeutic response. And the microglia correlate beautifully to lymphatic function dysfunction. And again, it's in a paper, right? We made those genes which are top 10, I think, % expressing genes, and you will see that many of the genes which we think are microglia genes are also expressed in lymphatic endothelial cells. And of course, many of the vascular genes, blood vascular endothelial cells, of course, are as well expressed in lymphatic endothelial cells. So I just think that we need to get to those lymphatic endothelial cells and then and understand what they express and how they behave in disease.
So I missed the beginning of the talk, so you may have already discussed the role of sleep in this story. So the glymphatic clearance system seems to be most active during slow wave sleep. And I wonder if you have studied sleep disruption and or enhancement as a way to manipulate the amyloid clearance.
Yeah, it's a very good question. So, I finally was able to recruit some really good postdoc who is actually expert coming from Wang Fang's lab. She was at Duke and now moved to MIT. So he actually finished with her his PhD and then came to my lab, and it's exactly what he's doing. So the question is whether sleep affects the function of lymphatic vessels and whether the improved glymphatic during sleep time is driven through improved lymphatic performance. And we have some preliminary data, but I have to keep it. So you invite me again, maybe in a year or two, and I'll present you that new data. It's a good question.
Thanks.
Yoni, question, if I could. So what's the missing element that's not clear if lymphatics are not intact? What is that species or those species? And why is it then lymphatics are necessary to clear those in the context of reduced plaque number? So what's special about what the lymphatics clear that is presumably activating microglia?
Oh, I don't think that it's a molecule that they clear. I think that microglia may well be just in this system, in this case, as be overloaded with clearance, right? So my understanding, and I could be totally wrong, but the oligomers are a very toxic form of amyloid beta, right? Am I correct?
Yes.
Yes.
Lots of evidence for that.
So if the sink is not functioning, and then there is and then the perfusion through the brain is very slow, was completely impaired, then even if you digest those plaques and then you release those oligomers, then they will be around in the neighborhood, and then they can potentially create the neuronal degeneration. So microglia activation may not necessarily be because of a lymphatic dysfunction that that does something to the plaque. It just may be the oligomers which cause degeneration or cause blood-brain barrier dysfunction. So we actually see the position. So I think this is staining for fibrinogen. So you see that in the control mouse with dysfunctional lymphatics, you see a lot of fibrinogen, which you do not see when we treat them with VEGFc. So again, you have dysfunctional blood-brain barrier, which Katarina Kasagalo showed very nicely, that also leads to microglia activation. So I just think that it's a very unhealthy tissue when there is no perfusion through the brain. That causes microglia activation, which potentially can, of course, cause many other secondary problems to the brain. So I don't really know what is the chicken and what is the egg in the system. It's a very complicated system. So we have now a new work also where we're actually showing how the function of lymphatic directly affects the function of blood-brain barrier. So could be that in this case it has nothing to do at all with the plaques. Is this functional lymphatic interferes with BBB function, and that's what drives microglia activation, which then so the plaque clearance is totally independent of this other pathologies that are happening.
Right. So the issue. So I guess the argument is it's not that there's anything special about what's in the brain. It's that the level is significantly higher, perhaps, in fact, as a result of plaque clearance.
Yes. Very well. Yeah. Absolutely. Totally. Yes. So again, I see this lymphatic as a sink of the brain, and if the sink is clogged, fixing the faucet is not going to help, right? You need to go to the problem in the plumbing.
Okay, well, let's. We're at risk of taking the rest of the hour talking about Alzheimer's disease, which of course is very much on topic, but. I'd love to hear the unpublished work.
Okay, let me do it very quickly. Okay, so we were very interested to understand this immune cells in the borders of the brain. And as I told you, it's very diverse environment, very diverse immunity there, and we did not know whether there is anything unique about them or not. So Andrea Kogura, who just defended, was a student in the lab, and then Toniki Mamuladze, who is a student in the program here, and Justin Assenhoven is a postdoc in the lab. They kind of tackle this whole this project together, which so I'll do it very quickly. It was published earlier. So before I get to the data, it's not published. So if we put two mice in parabiosis and we're looking into green cells in within the black mouse or wild type mouse, if we're looking at T cells plus minus, there is no much difference if we're looking into the blood and comparing them to different tissues like spinal cord dura or cranial dura, etc. But if we're looking at monocytes and neutrophils, what we see is that in the periphery, there will be about 30% of green cells in a normal mouse, but only less than 10% of green cells in the meninges of the mouse. And we said, okay, why is meninges not replacing a par with the blood? I mean, this is a 2 months parabiosis. So are these neutrophils and monocytes long-lived in the meninges? Well, the answer is no. Do we have precursors in the meninges? Well, the answer is no. Then what? How do you maintain population of myeloid cells in that area if they're not coming from the blood, where that's what we know from textbooks, monocytes coming from the peripheral blood? So a few years ago, 3 groups actually defined those channels which connect skull, bone marrow niches to the dura. As you know, skull is a flat bone, has lots of bone marrow in it. And so all this work showed that these skull bone marrow niches can provide immune cells, neutrophils, and here was lymphocytes, when there is a pathology. So the first paper was in stroke, neutrophils were coming, and second paper was lymphoma was using these routes. So we thought, okay, maybe this skull also provides immune cells constantly under homeostasis. Now, how do you address this question? How do you prove it? We can, of course, do transplantation of bone marrow and do chimeric mice and protect the head. I mean, and we did all this, and it's in the paper. But it suggests, but it doesn't really prove 100% because you can never really fully ablate the one aspect of the bone marrow versus the other. So then we said, okay, why not we cannot inject dye here because dye will leak as well. So we said, why don't you just replace a piece of skull? Why don't you take a mouse, wild-type mouse, remove a piece of skull and flap onto it a piece of skull from GFP mouse, which is an area which is kind of just above the confluence of sinuses, which is very enriched in bone marrow, which we did, and it worked because the cells were green here. After you can see after 30 days, there's still enough of green cells. Even after 2 months, there is enough of green cells. Now, we do not see any of these green cells in the periphery, very, very rare event. So therefore, it tells us that if we now see the green cell anywhere else, this green cell is coming from skull bone marrow niches. So indeed, if we're looking underneath the dura of transplanted mice, you can see here after 7 days, lots of green cells. And after 30 days, you can still see a lot of green cells. So we were focusing most on monocytes and neutrophils, but we also saw some other cells, B cells and DCs. And then we were working back to back with Marco Alona's group. So Marco actually showed the B cell aspect of the story, and we focused on myeloid and DC and neutrophils part of the story. So the cells are coming through those channels. You can see the channels sticking, basically going all the way from the dura into the skull bone marrow niches. And the immune cells are coming, as you can see here, they come not within, so you see, so blood vessel goes within the channel goes around blood vessel, and then the cell is coming not through the blood vessel, but basically in between the vessel and the channel. I don't know if you can see it here. So here the cell is, as you can see here, the cell is in between the vessel and the channel. Anyway, so this vessels or this bone marrow, of course, also is in vertebra. In vertebra, we found also channels, and the channels become very, very active if we induce an autoimmune disease EAE in mice. If we now make 2 mice together, do spinal cord injury, and then looking into one mouse, how many cells are coming from the other mouse, and we can see, for example, here, blood has about 25% of this one donor into the other, whereas spinal cord injury has about 10. So roughly 1/3 and 2/3 of cells. So majority of the cells in the injured spinal cord are coming from local sources, not from the periphery. In optic nerve injuries, even more so, we have almost 80% coming from local sources and only about 20% coming from the periphery. If we're doing skin injury, then there is not much difference between the periphery and local sources because, of course, skin doesn't have any underlying bone marrow next to it. If we're comparing the cells, so we can look at the cells which are enriched from local sources and cells which are enriched from periphery. Again, there is no way to do it pure because we cannot replace the entire skull. So here we're just doing a BMT without going too deep into this to save some time. What we're showing here is that cells that are in population of cells, this is EAE mouse, we see similar results in spinal cord injury. Cells that are coming from local reservoirs, from the bone marrow, skull bone marrow niches into the meninges and then into the injured tissue. These cells seem to be less inflammatory and more tissue building than the cells which are coming majority from the periphery, from the blood. And actually, there was a paper that kind of identified the phenotype of this very aggressive monocytes in EAE, and we see the signature to be enriched in cells which are coming from the periphery versus from local sources, which makes sense. Now, here comes the very interesting and new findings. So these 3 guys, so Jose is a MSTP student in the lab, Leon Smythe is a postdoc together with Justin Astenhoven, who also is a postdoc. What they were doing is they were doing spinal cord injury, and were taking the skull bone marrow niches because we wanted to save some animals, and so let's just with spinal cord we can use their bone marrow just for our other reasons, because so far away probably won't be affected. Well, actually, it was quite substantially affected, and we actually looked at all the progenitors in the bone marrow niche in the skull were altered by spinal cord injury. So at first we said, okay, how come the signal comes from such a far distance, right? And then Jose had this idea that maybe CSF is traveling, and CSF can maybe actually reach into the areas of the bone marrow niches, which sounded a little bit crazy. So they did experiments. So what you see here, this is a skull with the dura decalcified and then whole mounted. And so in this animal, we injected tracer into the CSF. And now we can look at different layer because it's a whole mount, a different layer, and see. So what you see here is this is the dura. You can see blood vessels in green and tracer in magenta. And then we're going higher from the dura, we're flying up through the channels. And you can see that in those channels, so blood vessels going through, but then between the blood vessel and the actual channel, you see the tracer. And then when we're reaching the bone marrow niches, you can see tracer also in the bone marrow niche. suggesting that CSF is going all the way from the CSF into the bone marrow niches and delivering the signals. You can see here we're flying up. We're now going through the channels. You can see the tracer is coming up as well. And then we are reaching the bone marrow niches, and you can see how bone marrow niche is very nicely labeled here as well with the tracer. So we also showed that this bone marrow niche is not only on the top of the skull, but of course in the bottom of the skull, which is known in humans and also in mice. You see these three beautiful reservoirs also have channels and are also labeled by CSF injection. So once you put anything in the CSF, it will cover the entire bone marrow throughout the whole skull, which is quite exciting. And in live imaging, we can also see that it's not really a phenomenon of a dead mouse, but also in live imaging, you can see the tracers are reaching all the way to bone marrow niches. So here is an example when we take a normal wild-type mouse, we're injecting the mouse into the CSF with antibodies against c-KIT, which is a marker on progenitor cells. So we're putting these antibodies into the CSF, and 100% of progenitors will be labeled in the skull, and nothing will be labeled in the tibia. So basically, CSF again delivers these antibodies all the way to the bone marrow niches. And even more crazy experiment, when we put these antibodies inside the brain, injecting it deep into the brain, with the glymphatic, it will come out into the CSF, and then will label about 50%, 70% of the bone marrow niches in the skull. So basically showing that whether molecule is in the brain, any molecule that's in the brain, once it leaves this into and gets through glymphatic into the CSF, it will be able to reach these bone marrow niches and actually alter them, and I'll show you in a second. I must show here that we were almost scooped, or actually were scooped by Matthias Nierdorf's lab, who showed very similar yet very different studies. He is looking at infections and shows that infectious agents, I think in this case was pathogens, which was viruses, which can also go from CSF directly into skull and bone marrow niches. So this is another experiment. So here we take a spinal cord injury. We're doing spinal cord injury. We're taking CSF from the injured mice, and then we're transferring them into a normal wild-type healthy animals. And what we see is within three hours, we see a very nice rush of monocytes directly from bone marrow niches into the meninges. So the chemokines within this CSF are calling the monocytes from bone marrow niches. Of course, there is a lot of potential, very interesting ideas here, because first of all, if there is a problem in the brain, whatever the problem is, it will be delivered to bone marrow and to local immune niches, and will either call the immune cells or may also alter the niche and potentially have some long-lasting effect on the entire immune response to the brain. But okay, I'll stop here. I'll thank, of course, all the funding sources and many collaborators. So all the Alzheimer's work is done in collaboration with Dave Holtzman, Carlos Kuchaga, Oscar Harari, and Celeste, and Bruno, and many other people here at WashU. And then the skull, bone marrow, we work in collaboration with Marco, and also with Dave, and of course many other people that I haven't had a chance to tell you what we're doing with them. Some collaborators from outside, and of course lab members. And I think I mentioned people as we were going through. So I guess I'll stop here and I'll take any questions.